remember that mitosis is the topic that you dealt with in grade 10. Now, mitosis occurs in every living organism in the somatic body cell to produce genetically identical cells. To mitosis lena yenza with body cells, not with sex cells. Okay, that is lot of body cells, we also mean somatic cells. Now, two identical cells are produced with identical chromosomes equal to the original cell. In mitosis, there's only one cycle, that is no 1 and 2 as in meiosis. Now we are having again this one cell. Let me rather choose a much clearer color here. So we're having this one cell here. This cell is going to divide. When it is dividing, it's going to form two daughter cells. Let's do this here. It's going to form two daughter cells. Here are the two daughter cells that we're trying to draw there. The two daughter cells, and these two daughter cells, it is clearly saying that they are different. They are the same with the parent cell. So another definition by Zanzi in mitosis, it occurs in every living organism. Two identical cells are produced with identical chromosome number. What actually do we mean by identical chromosome number? If we had here 46 chromosomes in this original cell, this simply tells us that even here we're going to have 46 chromosomes. Even here we're going to have 46 chromosomes at the end of what? Of mitosis. That means if we had a diploid, it's gonna, it's a small letter n there. If we're having a diploid number of cells, even here it's still gonna be a diploid number of cells like that, as well as here it's gonna be a diploid number of cells. So we're having those two ends to ends to ends, unlike in meiosis whereby the original cell had two n and then the daughter cells had haploid cells. So here everything is diploid. So there's only one division here. Asezobanayo second mitotic division. Jemba meiosis sinobani, no first and second meiotic division. So only here we are having one division, that is in mitosis. Now let us move now to another slide. The process of meiosis. It is a type of cell division that occurs in 4 daughter cells, each would have the number of chromosomes of the parent cell. Meiosis is a continuous process, but the events are divided into different phases. Okay, it's a continuous process. A process eyenzeka oko. All right, but njengoba isenzeka oko ayenzeki as soon as umntwana ezalwa intoni meiosis. It only occurs in meiosis xa umntwana reaches the stage esibizwa ba intoni puberty, and then from puberty it's going to be a continuous process. Okay, a continuous process, but the events in this continuous process are divided into different phases. All right, are divided into different phases. All right, events that are occurring in the interphase, those are the events that are occurring in the interphase, that is the DNA replication and the chromosomes becoming double-stranded, which were single-stranded. So this one occurs in the interphase. We must also bear in mind what facilitating interphase, facilitating a stage of what? This is not a stage of meiosis. They believe that interphase is the first stage. It's not the first stage. Interphase, it is whereby the cell has not yet divided, but it is preparing itself for dividing. So the stages of meiosis, they don't start in interphase, they start by prophase 1, right? So you must also bear that in mind, that the interphase is not the phase of meiosis, but it's the stage whereby a cell is dividing, is preparing itself for division. All right, now we move on to the next slide. The next slide, each chromosome will now consist of 2 gembastays are double-stranded, and then the chromatids joined by centromere. Ngala process ye-DNA replication nansi ke ngesezantsi uba yenzeka kanjani DNA replication, ukuyititshiwe ke-DNA replication kwi-DNA code of life. But the DNA replication helps to double the genetic material so that it can be shared equally by the new cells arising from cell division. So that's the DNA replication. But this way, it's not how it is written to describe the process of DNA replication. Apha uyayidefinelwa lapha uyaxela uba yenza ntoni, awuxelelwanga uba how does it take place. So how to describe, a question to describe usually ayidli ngaba na 2 marks. Iba zisa questions zidla ngoba no 5 marks, 6 marks, 4 marks, 3 marks, and so on, so on. It's a different question to describe. Ayifani nabani notify. All right. Then these are the phases of meiosis. Naziya zivelile phaya p-met, that is prophase 1 and 2, metaphase 1 and 2, anaphase 1 and 2, telophase 1 and 2. But this is right. It's not prophase 1 and 2 in a correct order. It's prophase 1. After prophase 1, metaphase 2. Prophase 1, metaphase 1, anaphase 1, telophase 1, and then level of phase yoqala, that is this one. Esiqala ngayo apha. Let me do this. This one. This is meiosis 1. This one. This is meiosis 1. And then this one. This is meiosis 2. Okay. So meiosis 1 simbona ngoba unaba 1 phaya, those 1 there, and so on and so on. But meiosis 2 simbona there's Roman figure phaya. Remember meiosis 2 as the symbol as 2 like that. We write just simple that Roman figure there. Okay, so those are the stages of what? Of meiosis. Now, what is happening there on this diagram on this side? On this diagram, what actually is happening? This diagram on this side. Let me try to make this highlighter a little bit bigger. Yes. What is happening here is the process of what? Of meiosis. Remember, we have said that meiosis into zophume are 4 daughter cells. Do you remember? Starting as 4 daughter cells, if the meiosis yenzeka lapha, yenzeka in plants, and the plant is a male, as 4 daughter cells phaya kuzoba the pollen grains okanye pollen. If yenzeka kwi-female plant, these are going to be the ovules in the ovary. If yenzeka kwi-male, a human, these are going to be sperm cells. If yenzeka kwi-female, a human, these are going to be egg cells. Okay, of course, egg cell, these 3 are going to disappear maturation. Only this one libe one iqanda elibanjani elba full matured. Okay, so this is the process of meiosis. Here we can see that this is meiosis 1 from here to okay, sorry about this. Let me try to use this one. I think it's gonna be a little more better than that one. This one, I am not sure what's happening here. This one is meiosis 1. What is happening there? And then we're having meiosis 2 here. Okay, so this is meiosis 1, this is meiosis 2. You can see that at the end of meiosis 1, we're having 2 daughter cells, but these 2 daughter cells, they continue dividing for meiosis 2, and then meiosis 2 will end up having 4 daughter cells at the end of all this. Now we go on to the next slide. The next slide shows us what is happening in meiosis 1. Remember that we have said meiosis 1 is a first meiotic division, and it is called a reduction division. When we talk of reduction division, let us highlight that term there, reduction division. That means a reduction coming from the word reduce. That means apha kuya reducwa here, ne? Into le reducwayo the chromosomes, where the number of chromosomes in the nucleus is halved. So go reduce chromosomes. The chromosome number kanjani? By halving it. Okay, that means if at the start of meiosis you are having four chromosomes. Okay, then what will happen at the end of meiosis? Sitemo said this is halving, right? What will happen at the end of meiosis? Remember we've said we are doing what? We are halving. So if we are halving at the end of meiosis, we need to get, let me put an arrow there. Yes. Now at the end of meiosis, you need to have what? 2, because the half of 4 is 2. That means once again do only meiosis. But if the mitosis leyenzekayo, ne, bese zobana how many chromosomes? Initially, let's say it's the chromosomes as 4. Now these 4 chromosomes, they undergo mitosis in one cell, ne? The cell na 4 chromosomes and undergo mitosis. This is how many chromosomes at the end of mitosis? Simply still going to remain 4 because in mitosis. Now the reason it means this is because the half number of chromosomes at the end of what? Of meiosis. Okay, meiosis 1 rather. Now prophase 1 of meiosis 1, we're saying the chromatin network condense. Okay, the chromatin network condense. When we say that chromatin network condense, we are saying the chromatin network yathini iyamaturisha, ne? Iyamaturisha iformisha ngentoni chromosomes. Siyasuka from being chromatin, so there's a chromosomes. Hence you can see here we are no longer having the chromatids, we're having the chromosomes. Again, they are saying homologous chromosomes are formed. Now, homologous ngaphambili, what did we say about homologous chromosome? We have said that homologous chromosome, okay, there's something that's disturbing me here. We have said that homologous chromosomes, if these chromosomes like these, they pair like that, these 2, the first one, these chromosomes, the other one is from the paternal, that is from the father, and the other one is from the mother, which is maternal. So they are there standing together as partnership, the homologous pair. At the same time, this is another homologous pair of chromosomes. So they are now in prophase 1, so homologous pair. And then crossing over takes place. Now, again, crossing over. Crossing over, when they're done, it's whereby we're crossing over. Then we want these 2 homologous pairs of chromosomes here. 2 homologous pairs of chromosomes. What's going to happen is that this chromatid for this one will overlap to the chromatid of this one. So it crossing over yenzeka here. This is where crossing over takes place. Okay? This chromatid overlaps on this chromatid and then passes the genetic information from this chromosome to this one. And at the same time, this one passes this chromosome, this genetic material from this chromosome to that one. So it's a signal process in Done is crossing over. And the point is to exchange this information. Siyibiza ngoba yintoni? Siyibiza ngoba yi-chiasma. Kanye siyibiza ngoba yintoni? I-chiasma. Let me try to write these terms here so that izokwazi uba zibhalwa zizazo zithini. Sithi kwenzeka ntoni phaya? Kwenzeka i-crossing over, right? Yenzeka apha crossing over. Yenzeka crossing over at the point esiyibiza ngoba yintoni? It's a chiasma, right? This is the location, a point where crossing over occurs, right? Remember, the crossing over, it serves as a variation, all right? So this is called a chiasm at that point. Now we're saying the spindle threads start to form. The spindle threads there, as you can see, those are what as spindle threads. Okay, which are going to pull the chromosomes in opposite poles. And then lastly, nuclear membrane disappears. Bekade kuyi-nuclear membrane layer uyibone ngathi disappearisha phaya. So asizuba nayo ngentoni i-nuclear membrane. Okay, so that's what actually happens on what? On prophase 1. Now we go to prophase 2. Sorry, so we go to metaphase 1. Oh, but before we go to metaphase 1, let us first start moving from this prophase 1. Explain what actually happens during crossing over that I've already explained. Okay. Homologous chromosomes align together, as you can see over there. They are aligning together. One chromatid of each chromosome overlaps with a chromatid of its homologous partner. Chromatid overlap there. Okay. Then a crossing over takes place at a point called chiasma, as I've just mentioned earlier. Okay. The chromosomes separate in such that each has new genetic material from both chromosomes that is shared. After once again crossing over, we are now having these chromosomes here. I'm trying to put this thing here. Now we're having this chromosome here and another chromosome there. All right. When you look at there, you will notice that these chromosomes, before, let us first take this one. On this portion, it had A, B, C as was. Okay, let me put it like this. It had that C as a what? As a capital letter. Okay. And then the red one, on this one, it now brings this C as a what? As a small letter after crossing over. Okay. And then this one, the red one, had a C as a what? As a small letter. Okay. But now after crossing over, that C is no longer a small letter, it's now a capital letter. What does that mean? This means that this C from this chromosome exchange, or this gene from this chromosome, was passed on to this chromosome. And then at the same time, this gene for this small letter C and this chromosome was passed to this chromosome. So the chromosome iphethe information enayo the information yangapha. At the same time, that this one iphethe information enayo the information yangapha. So if this one was from the father, and this one was from the mother. Now kulesiwa from data sekhona portion kamama, kulesiwa kumama sekhona a portion kabani katata. This is the result of what? Of the crossing over. That's what they were explaining here on these notes on this side. Okay, so this diagram down there, you'll notice that homologous chromosomes, there you are, you can see them. These are homologous chromosomes, and then crossing over takes place here, and then after crossing over has taken place, the genetic material has been passed on to each other. Okay, now we can move to the next slide. That is now the second stage of metaphase 1. Now in metaphase 1, what you need to take note of is that in metaphase 1, the chromosomes now, they are aligning themselves at the what? At the equator. Right? You can see that the chromosomes now, they are at the equator. Okay, they are going to align themselves like that at the equator. Hence the term matter, that ME, you will remember it. M for middle. Okay, so this is phakathi pi chromosome, a homologous pairs of chromosome, because this is metaphase 1. Zisuka kubani ku homologous pairs bekubani beprophase 1. Okay, now we are saying homologous chromosomes randomly line up along the equator, as you can see there, of the cells in 2 rows. Then this row and this one, this one and this one. If in time is 46, the next one will be there and there, then so on. Each chromosome is attached to the spindle thread from each pole. As you can see, the spindle fibers as it develop prophase 1. Nazi is attached to our chromosome. So it's to the what? To the pole. Okay. So this is what actually happens. Simple into metaphase 1, and then we go to anaphase 1, the third stage, anaphase 1. So ana, there's that A there, meaning that away. Now the chromosomes, if you can look clearly now, the chromosomes there, they are being pulled away. You can look there, you see now, they are being pulled away. They are being pulled away. Excuse me here. They are being pulled away. Look, chromosomes there. You can see the shape. It's like it centromere. And where is the direction of being of pulling going? The direction of pulling is going this one, Gulena, is going that one, Gulena, is going this way, Gulena, and to this one is going that way. That means the sentence of sharing a code, right? So this is anaphase. The spindle thread shorten, shorten and pull the homologous chromosomes to the opposite poles. Then the second bullet is that chromosomes of the homologous pair separate. So la homology or partner bekhona phaya ihlukana enye ngapha enye ngapha ezinye izilandelayo njalo. So the homologous chromosomes, they are being pulled off like that. They are separating. Okay, and then we go to telophase 1. Now we will have in fact the cell has finished now to pull, the spindle fibers is finished to pull. Now it's no longer pulling. These chromosomes that were pulled there, they now reached the poles, and what is going to happen is that the cell is going to divide through the process ebizwa yintoni i-cytokinesis. Libalulekile ke lagama abantwana baye balibale ungathi it's not important. Through the process of cytokinesis. What is the process whereby the cytoplasm and cell membrane or during telophase 1 divide? You are going to say it's what? It's cytokinesis. Then we'll remember that we're going to diploid cell, and it two haploid cells now. Okay, so we've got 2 haploid cells. All right, these 2 haploid cells, it would be good to point them. There are they. This is the first one. This is another one. There's a guard as 10. There's a guard as the sensor cell, Z cell A1. Let us try to draw that cell there. There's that is cell A1. Prophase 1, metaphase 1, anaphase 1. Now, this is telophase. Yes, split and a cell. Split up. The splitter is paratin. The cell splits like that. And then el-qala lizengapa, el-qala lizengapa. Okay? All right. So you need to put that in mind. That let me put this one like this and try to show that arrow there a little bit, yes, on the side. Okay, so now we're having 2 cells. This one is labeled at the file ayifanga phese but isplithe phakathi yanamacala ayi-2. So akukho 3 cells there. 2 cells in telophase. Then the nucleus reforms. The nucleus disappear nucleolus rather. Nucleolus reforms disappear during prophase 1. Yeah, reform again. Iphinde bekhona new nuclear membrane, nuclear membrane again. So this is what actually happens in telophase 1. Now let us go to meiosis 2. Meiosis 2 starts by prophase 2. Kwenzeka ntoni prophase 2? The nucleus and nuclear membrane disappear. Sorry, not nucleus, nucleolus. Le nucleolus yaphinde yafoma telophase 1. Disappearisha again kubani prophase 2. Now the nuclear membrane, they disappear. Okay? And then the spindle fiber are formed. Phinde kuformisha a new spindle fiber ebezingekho kubani telophase 1, and then chromosomes become visible. Now, don't forget uqaphela lapha ku-meiosis 2 kuba njengoba ku-meiosis 1 bese ne-cell ebiyi-1 kwenzeka ku-prophase, metaphase, anaphase kuyo, then once sifika ku-telophase 1 zayi-2. Now zezi cells eziyi-2 ezibize kubani beze ku-telophase 1 zizoqhubeleka ngendlela efanayo. Okay, so that is to say le cell E1, this one, let me try to draw it so that uzokwazi uba uyibone. Alright, in fact mandifake nje arrow. This cell, this one will undergo the same process as this one. Okay, will undergo the same process as this one. So if prophase 2 here, it's going to be prophase 2 here. Okay, so notice this thing. Now we no longer have homologous chromosomes here. Okay, and we're no longer having a crossing over here in prophase 2. Okay, that's why ungazibona ezo zinto zibhaliwe prophase 2. Nesithetha ngayo prophase 2, i-nucleolus ne-nuclear membrane ziyadisappearisha, spindle fiber ziyafomisheka, chromosomes become visible, not homologous chromosomes. Bear in mind. Okay, metaphase 2, individual chromosomes line up at the equator. Look at that. These chromosomes are individual. They are not homologous as in prophase 1, right? As in metaphase 1 rather. So here is just single chromosome, single chromosome. Now let's say single chromosome, single chromosome. As a partner, partner Y chromosome there. Okay, so that's one other difference between metaphase 1 and metaphase 2. Individual chromosomes line up at the equator of each cell with the centromere attached to the spindle fiber. So now kubandwa this centromere and then this centromere on this side, and then a spindle fiber so the centromere phaya esinye sibambe kwacala ngapha. Okay, I hope it's becoming clearer and clearer. And then we're having anaphase, whereby the spindle fibers start to contract. That is now anaphase A, stay away. Again, is push away. But what we need to take note of, anaphase 1, the whole chromosome in anaphase 1, the whole chromosome. But here it's just what that chromosome A1 is split apart. Right, because the chromatid on this side, because the chromatid on that side, and then other chromatid on this side. Okay, same applies. Now let's say the same thing. And on that one, the chromatids, not the chromosome. So the chromosome break apart. When the chromosome breaks, what's going to happen? Chromatid now we're now having z-daughter chromosomes. Okay, because chromatid centromere, you must know that's a chromosome. Okay, so these are daughter chromosomes. Okay, so that is what is happening in anaphase 2. This is what is happening in anaphase 2. Let's perhaps go to telophase telophase, let me try to put this thing here, telophase 2. What is happening in telophase 2? The daughter chromosomes reach the poles. With daughter chromosomes to the poles. Ne? After that, a new nucleus forms. Lanti bisenzeka ku-telophase 1 yenzeka again lapha, but umehluko ku-telophase 1 besithetha nge-chromosome, not nge-daughter chromosome. A new nucleus forms. The cell membrane of each cell constricts. Bonal cell membrane. The cytoplasm divides into 2 cells. Cytokinesis again. Now, instead of 2 cells, ebesenza ku-telophase 1, now 4 haploid cells. Outer haploid. I want us to see this haploid. Haploid. Look at that. Initially, in metaphase 1, in prophase 1, we had how many chromosomes? 1, 2, 3, 4 chromosomes, right? And sitting one lona, this is diploid, that is 2N. So who's being haploid? 2. Now let us go to this stage of telophase 2. Now we're having 1, 2, 1, 2, 1, 2, 1, 2. That means it's 2, 2, 2, 2, meaning it's the half of 1 of 4. So this is now no longer diploid, but it is haploid. That's why we have 4 haploid daughter cells. How many cells? 1, 2, 3, 4. There are 4 haploid daughter cells there. The daughter cells are genetically different from each other. When you look here, this chromosome is dark there, and then it's bright there. It's really not bright pelele. Also, if you cell as one chromosome is this one is the vice versa. It's bright, a little cool, dark. And bright and dark and bright. So they are different from each other. So it was the daughter cells are genetically different from each other. Then that's the end of telophase 2. Xa uphela telophase 2 kuthi ngokuphela intoni imeiosis, yenzeke yonke intoni imeiosis. Now nantsi enye into ebalulekileyo ekufuna uvule ufunde uboya rhalela uvele uyinqaye. Okay, the importance of meiosis. Ibaluleke ngantoni meiosis? This is one of the requirements ezikhona exam guideline. 1, production of haploid gametes. Lesigqiba kuyenza. Okwesibini, the halving effect of meiosis overcomes the doubling effect of fertilization. Ithetha ukuthini le? As a statement, the halving effect of meiosis overcomes the doubling effect of fertilization. If bekuthiwa azihalfisheki, zihlala zingu-2N njengoba i-original cell bangu-diploid. If bekuthiwa ke ne-gametes ezifomishekayo ziba ngu-diploid, kutsho ukuthi xa kusenzeka i-fertilization, where's my pen here? Kutsho ukuthi xa kusenzeka i-fertilization, it was going to be diploid. Okay, lo diploid ngowe-sperm cell ahlangane no diploid we-egg cell, and then bazokhupha izygote ke ngoku u-2 plus 2 ngubani? 4, and that means N. So that means izygote bizwa ngu-4N. Ikhule lo zygote ngu-4N, and iphinde ihlangane nenye ngu-4N nayo. And meiosis are you having effect? That means la 4N ebezophinda hlangana no-4N. So kutho ukuthi thina besohlala sinezinto ezonyukayo, i-number of chromosomes ezonyukayo. So imeiosis ke ithi no, kunoba ke ngoku ukhule ngolu hlobo, kungcono sithi meiosis ihaflishe la 2N xa kuzokwenzeka i-gametes abe ngu-N no-N. So N no N uzokwenzani? U-N, what's that N going to do? Uzo-edisheka nomnye ubani? Nomnye N. Uzo-edisheka nomnye N, and then kube sekuphuma bani? Kube sekuphuma 2N olingana ne-body cell. That's why ke meiosis ikhona. So as the statement asithi sorry, let me clear here and then try to move this one. As a statement esithi the halving effect of meiosis overcomes the doubling effect of fertilization. Sinyanisile because uba bekuthiwa ayikho la halving effect ye-meiosis, fertilization izohleli idablisha thus maintaining a constant chromosome number of one generation to the next produces genetic variation by way of the process of combination. Why is meiosis important for survival? Pendula uthi, it is of great importance because it creates genetic diversity in the population. That means yenza uba kube khona different kinds of that kind of organism on the meiosis kuyo. Umzekelo ungathi ngenxa yoba umde uzala abantwana abade bodwa bafana nawe, so ungakwazi ukuba nabantwana abafutshane, ungakwazi ukuba nabantwana abanomzimba omkhulu nomzimba omncinci. So yonke loo nto is because of intoni? I-meiosis. Hence difference is in survival of organism in environment. So i-meiosis yonke yenza loo nto, variation. Xa sithi variation sithetha ngantoni? Sithetha about the differences between organisms of the same species. That is, xa sibhekisa statement singabantu mos, okay? So variation abantu ndithetha ukuthi khona abantu abamnyama, abamhlophe, amakhaladi, abemlungu, what, yi-variation that one, ne? So it may be due to genetic factors or environmental factors. Into engayikhoza loo nto babe khona abantu abamhlophe, abamnyama, abade, abafutshane, abantintini. It's due to genetic factors okanye environmental factors. What are the causes of this genetic variation? One, it's crossing over. Let's take ngayo. Two, random arrangement of chromosomes during metaphase. Phaa metaphase, masiqala metaphase 1, ungavula i-study guide sakho ujonge mind the gap wakho okanye whatsoever textbook you're using Africa. You can open your mind the gap, your understanding whatsoever textbook or study guide you have. You'll notice that during metaphase, the chromosomes are arranged at the equator not as single chromosomes, metaphase 1, as homologous pair of chromosomes. Okay? Now, as they are arranged there as homologous pairs of chromosomes, I want us to go back there. They are arranged there. Now, a random arrangement, iti le chromosome ngapha ibisenoba kwecala lena ibe ngapha. Mhlambe ezi zihlale zinje okanye zihlale zinje ezi lena ibe ngapha, lena ibe ngapha. So that sekupholishwa ngu-anaphase ziyahamba, kungahambi lena le, mhlambe kuhambe lena le zize kule pholi ngapha because bezobe secaleni liyi-1, lena ihambe nalena zize kule pholi ngapha because bezobe secaleni liyi-1. So and apha ke ngokuphuma something eyenye engafani nalena ephumileyo phayane. So that's random arrangement of chromosomes during metaphase. Metaphase 2 nakhona ungenzeka the same thing. Instead of the chromosomes zime kanje lena iguquke le chromatid ngapha bengapha, now lena mhlambe ngapha, then again this daughter chromosome ngapha nalena ikwecala. So that's what that statement means. City random arrangement of chromosomes during metaphase. Thirdly, random fertilization. Akunyanzelekanga uba ibe sperm cell esithile esizofertilize iqanda elithile. So any sperm cell can fertilize. Okay? Random mating, akunyanzelekanga uba i-female ethile mayidibane ne-male ethile. The male can choose any female to mate. Okay? From abantwana whatsoever abantwana baphumayo. Then lastly, it can be mutation. Yintoni ke mutation? I-mutation zii-mistakes, zii-errors eziye zenzeke xa kusenzeka intoni? Xa kusenzeka i-meiosis in this case, ne? So kungenzeka kule cell ingakwazi ukubreaka kakuhle xa kusenzeka i-meiosis. So a mutation, that one. So it can lead to intoni? A variation. Okay? Causes of variation nazo ezicacisiwe phaya ngale ndlela bendicacisa ngayo, ne-random arrangement of chromosomes there. You can read them on your own. Random fertilization, random mating. You can see phaya eziyabhitlezi phaya ingamitha noba yeyiphi kweziyabhitlezi phaya. And then lastly, number 5, we're having mutation. Changes nazi mutation, kwenzeke mutation phaya kulaa ndawo, instead of ukudibana mhlawumbi ii-bases kulaa codon, In fact, it's not a codon in this case because the DNA, DNA triplet code there. Suppose something, that one instead of that one. So that's a mutation. So is a changing of the complete structure of what? Of a DNA or a protein whatsoever that is formed there. Non-disjunction and its consequences, right? Xa sithetha nge-non-disjunction and its consequences, it is whereby, okay, masiyifunde is the failure of chromosomes or chromatids to separate during anaphase. So singathi is the failure of chromosomes during anaphase 1 to separate, or is the failure of chromatids during anaphase 2 to separate. So chromatids was anaphase 2, or chromosomes were anaphase 1. Once separate chromosomes or chromatids during anaphase, izolithela loo nto kuthiwa intoni esiyibiza uba yintoni i-non-disjunction. Ziveziwe kakuhle ke lapho masiqale ngalena kwecala ne. Here this is masiqale ngalena ka-meiosis 1 yenze ku-anaphase 1. You see this homologous pair of chromosomes, it's supposed to break like that, form that close chromosomes and those chromosomes, okay? And then in meiosis 1, at the end of meiosis 1, we end up in telophase, but we failed to have that, okay? Instead of this chromosome to be pulled on that side, it does not allow to break. It fails to separate. So instead, it goes to this side. So you want a non-disjunction, that one. If there was a non-disjunction, and only nale disorder is of formation. In non-disjunction, it's not a disorder. Okay? In non-disjunction, it's a condition that leads to a disorder. Umzekelo, xa le non-disjunction, let's say the chromosome pair belapha being chromosome pair number 21. So yenzeka chromosome pair number 21. It was going to lead to a disorder esiyibiza uba yintoni i-trisomy 21 okanye i-Down syndrome. So there's a difference between a disorder and a condition. So non-disjunction is a condition whereby there's a failure of chromosomes or chromatids to separate during anaphase. Okay? So nanti-ke ifile useparate ihamba yonke la chromosome instead of going to chromatid A1. And then xa sesiphuma ngokugqibeleni gametes zalapha, instead of us beyi-2, sibe 1, 2, 3. Instead of us beyi-2 lapha, it's 1 here, and then up as normal on this side. Ne? So this leads to, if you have chromosome pair number 21, leads to a Down syndrome. Okay? And then on the other hand, the same thing, the breaker, but instead of this one, you have meiosis 2 with this one, a chromatid, I break this one. Okay? We'll move to the next slide. Each nucleus should contain 23 chromosomes after meiosis, but if non-disjunction occurs, one nucleus contains 22, while the other is 24. When either of these resulting gametes fuses with a normal gamete, they result in 45. They result in 45 or 47 chromosomes instead of 46. 23 plus 22 is 45. 23 plus 24 is 47. So, once again, non-disjunction. Here. So for this one, if this one fuses with a normal one, kuzophuma ena 1 extra. So it was a 46 higher plus this one is by 47. So there's 23. So plus this one being 24, you fuse on 23 from 47. Lena ya short, okay? So 20 must be 23, ngoku 22. So you fuse with a normal one, so must ngapi Z20. As a must Z40, Z45. Okay, so that's what this actually means. This may lead to Down syndrome child. The child will have 45 autosomes and 3 number of 45 autosomes. The autosomes are the chromosomes as is so from pair 1 to pair 22. Okay, with 3 number of chromosomes or 21 chromosomes instead of the normal pair of 1 pair of sex cells, of sex chromosomes rather. Women over the age of 40 have a greater chance of having a child with Down syndrome. Sesinomkeso-ke uba abantu abesimame selebe ngapha kwa-40 banee-chances ezininzi zokuba nabantwana abane-Down syndrome. Simbona ngantoni ke ngumntwana une-Down syndrome? The physical symptoms, stocky body, ufumanisa umzimba wakhe it's round, solid, ne? Ubonakala ngathi umuntu onomzimba oqinileyo omkhudlwanyana, ne? Distinct facial features such as flattened face, ubuso bakhe bu-flat with small broad nose, small skin, folds in the inner corner of the eyes that appear in the slant upwards. Bonakala ngathi amehlo akhe abungenhla kancinci, abuphezulu kancinci, asilandele ngasentla kancinci, ears situated ngasezantsi, iindlebe zisezantsi, and then ulwimi lukhulu, broad hands and short fingers. All this mixture that has been explained has been dotted down there. Now, another thing that is very much important, differences between meiosis 1 and meiosis 2. Now, as is straightforward, but meiosis 1 chromosomes arrange at the equator of the cell, and the whole chromosome move to the opposite poles. And now by chromosomes line up at the equator of the cell individually. Partneration, the chromosomes or chromatids move to opposite poles. 2 cells in meiosis 1, 2 cells form at the, just read them like that. I won't have to explain them. We've been talking about them. Similarities in mitosis and meiosis. Because the mitosis and meiosis, both processes involve division of nucleus. Both involve cell division, that is formation of new cells. Both the processes occur in the M phase of the cell cycle, that is before the cell divides there. In both cells, the stages are common. That is once again prophase, metaphase, anaphase, and telophase. The synthesis of DNA occurs in both. Cytokinesis takes place in both. Then now we're going to differences between mitosis now and meiosis. Differences between meiosis 1 and meiosis 2. Similarities in mitosis and meiosis. Many differences in mitosis and meiosis. Mitosis occurs in the somatic cells. Meiosis occurs in the gametes. That is it occurs in the sex cells rather, and not in the gametes. Right? It occurs in the sex cells. Production of genetical identical cells. Mitosis, meiosis, production of genetical non-identical cells. Mitosis results in 2 daughter cells. Meiosis in 4 daughter cells. Mitosis 1 nuclear division, meiosis 2 nuclear division. Mitosis no crossing over in prophysis 1, meiosis crossing over in prophysis 1 occurs. Mitosis resulting daughter cells are diploid, meiosis the haploid daughter cells. And lastly, mitosis results in somatic cells, in somatic cells. And the meiosis the gametes, that is the sperm and the sperm cell and the egg cell, which is the ova. So that is as far as you can go. So that's where we come into